Hello everyone and welcome back to my channel. Today I will be discussing about the structure of DNA. So the double helical structure of DNA was first proposed by Watson and Crick, and according to them the structure of DNA is made up of two polynucleotide chains, and the basic unit or the structural unit of polynucleotide chain is nucleotide. That is, a nucleotide will repeat in itself to produce a polynucleotide chain, and a nucleotide is made up of three elements: a 5 carbon sugar, or we can also call it a pentose sugar, a phosphate group, and a nitrogenous base. Now any one of these nitrogenous bases will remain present, either adenine or guanine or thymine or cytosine. Now in case of RNA, thymine will always be replaced by uracil. That is, there will be no thymine. In place of thymine, uracil will remain present. This is one difference between DNA and RNA. So here we can see that this appears like a ladder-like structure. Now if we twist this structure, we will get the double helical structure of DNA. Now due to the helical structure, we will find two grooves. The bigger one is known as major groove and the smaller one is known as the minor groove. Now these grooves are essential for the attachment of the protein for the purpose of replication or transcription. Now if we take a turn of the DNA, we will see that there are 10 nucleotides in each turn, and the length of each turn is 3.4 nanometer, and the distance between two nucleotides is 0.34 nanometer, and the diameter of the helix is 2 nanometer. So here we can see the arrangement of the elements within a nucleotide. This is a pentose sugar, this is a nitrogenous base, and this is the phosphate group. So as this is a pentose sugar, so therefore there will be 5 carbon. This is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4, and this is carbon 5. And at the first position, nitrogenous base will remain attached always. And there are two types of nitrogenous bases. This can be either pyrimidine or purine. Now those nitrogenous bases which have only one ring, these are known as pyrimidine. This is a pyrimidine. And those nitrogenous bases which will have two rings, these are known as purine. So this is a purine. Now cytosine, uracil, and thymine are pyrimidine, and adenine and guanine are purine. Now how to remember this? You can remember it by cut pi, and the remaining ones will be purine. And a pyrimidine will always form a bond with purine, and a purine will always form a bond with a pyrimidine. Now at the 2' position, in case of DNA, no oxygen will remain present. That is why this is known as deoxy. Whereas in case of RNA, oxygen will remain present. And this is another difference between DNA and RNA. And at the 5th position, the phosphate group will remain attached. Now as phosphate group is negatively charged, the entire DNA is negatively charged. Now this is the structure of a nucleotide, and now this nucleotide will repeat in itself to form the polynucleotide chain. So now this phosphate group will attach itself to the adjacent nucleotide by binding itself to the third carbon of the phosphate group, of the pentose sugar of the adjacent nucleotide. So now the bond by which it forms is the phosphodiester bond. Since two ester bonds are formed, this is known as phosphodiester bond. So this makes the sugar phosphate backbone. This is the sugar phosphate backbone. Now adenine will always bind with thymine with 2 hydrogen bond, and guanine will bind with cytosine with 3 hydrogen bonds. And the polarity is always opposite. That is, if one strand is from 5' to 3' direction, the other strand will be from 3' to 5' direction. Here we can see that this is the structure of the sugar, and here the 5th 5' phosphate group is open, and here the 3' hydroxyl group is open. And in the opposite strand, the orientation of the sugar group is completely opposite, and that is why we will find that here 3' OH group is open and here 5' phosphate group is open, and that is why the polarity is different. Now here we can see this is the entire structure of the DNA. This is the sugar phosphate backbone. This is the sugar phosphate backbone. This is formed by phosphodiester bond, and in between this the nitrogenous bases are present. Now we can see that adenine is binding with thymine with 2 hydrogen bond, and guanine is binding with cytosine with 3 hydrogen bond. Now this is very much essential that a purine will bind with pyrimidine. This is because, see, if suppose adenine is binding with another purine, then there will be 4 rings, and pyrimidine is binding with another pyrimidine, then there will be 2 rings total. So this will alter the diameter of the helix. So to maintain the diameter of the helix, a pyrimidine will always bind with the purine. So yeah, this is all about the structure of the DNA. I hope you will find it helpful, and if you like the video, please do like, share, comment and subscribe to my channel. Okay, bye. Thank you.